Good afternoon, everybody. It's my pleasure to welcome you to the 2022 seminar series of Quantitative Life Sciences and Medicine. This is actually our 6th year of the Quantitative Life Sciences PhD program, which is really exciting. And it's a great pleasure to have with us today Dr. Subramanian speaking today, and I'm going to invite Professor Mandel to introduce her.
Great, thanks so much. It's really a pleasure to welcome Naeha here today. Naeha and I go back to being postdocs together in Ron Germain's lab at the NIH, where we both sort of embarked, I think, on our systems immunology career, if you will. And I remember Naeha for having arguments with Ron and usually prevailing. So I think that tells you a lot about her science. So Naeha is an associate professor in the Department of Immunology at the University of Washington, and she's at the Institute for Systems Biology in Seattle. And she's really an expert in studying innate immunity, looking at inflammasome activation and using systems biology tools to do so. So I thought she would be really a fantastic speaker to bring in for this QLS series and share some of her latest work in that field. So the floor is yours, Naeha. Thanks so much again. I'm really excited to have you here.
Thank you, Judith. The pleasure is all mine. And thanks everyone for joining remotely. So like Judith said, we are really at the interface of innate immunity and systems biology. And I like to think that we're still, we started to use more and more of these big data tools in our work, but I like to think that our work is still rooted in the basic concepts of innate immunity. So my lab essentially is interested in this innate immunity triad where, we know that the innate immune system is absolutely required for host defense against pathogens, but if this is dysregulated, can lead to a variety of diseases. Can I check? Can you see my pointer, or do I need to?
Yep, we can see it.
Yeah. Okay. Cool. Okay, so I'll just go with the mouse pointer then. Yeah, so we know the innate immune system is absolutely required to control pathogens, but if this goes wrong, that is, if there's too little or too much of it, you can start snowballing into disease. And so for today's talk, I'll mainly focus on this first arm of host pathogen crosstalk, and I'm going to be ambitious and try to present to you 2 vignettes of recent work. This is all unpublished. So, and actually, incidentally, is the first time that I am presenting this. So you are all my test subjects. So feel free to test me and throw out as many questions. And we will see if our story prevails. So the 2 vignettes are going to be around studying Salmonella host pathogen interactions and some of our recent work on Lyme disease. So let's dive right into the first vignette on host pathogen crosstalk. So we've been interested largely in innate immune control of intracellular pathogens, mainly in the context of Salmonella infections. But before diving into the data, let me give you an idea about innate immunity for those who are not exactly in the field. And so the innate immune system basically senses a variety of microbes and danger signals. It's essentially your first line of defense against pathogens. You can kind of think of it as a burglar alarm that is going to go off when an intruder enters into your house, kind of alerting you to danger, and the immune system then responding by secreting a variety of cytokines and chemokines. And so this recognition or this detection is mediated by predominantly 2 arms, an extracellular arm, which comprises primarily of the TLRs, and then just a wide variety of intracellular sensors, the largest class of which are these NLRs, or knot-like receptors. And this is the class of proteins that we've been interested in. They are involved in detecting pathogens and danger signals, and mutations and polymorphisms in them have also been associated with just a wide variety of autoimmune and autoinflammatory disorders. A very interesting area, that's pathogen-independent induction of autoimmunity and autoinflammatory disorders, right? So that's a very interesting area in and of itself that I won't be diving in much into today, but be focusing largely on their ability to detect pathogens. And so the pathogen that we've been interested in, at least in recent times, has been Salmonella. This is really a model pathogen that infects the gut. Most of you would know that it's taken up in contaminated food and water and accesses the intestine, which basically is the first mucosal tissue that Salmonella infects. And upon doing so, it infects these intestinal epithelial cells and M cells, which are these more highly phagocytic macrophage property-like epithelial cells. And eventually, it goes systemic into the blood. But upon first infecting intestinal epithelial cells, Salmonella is taken up by myeloid cells, within which it activates Caspase-1. Now, as Judith said, my lab has been interested in inflammasomes, and so Caspase-1 activation is mediated primarily through inflammasome activation, resulting in production of IL-1 beta and IL-18. IL-1 beta is a really potent recruiter of neutrophils. And this early recruitment of neutrophils is actually required to clear Salmonella infection. But Salmonella has actually evolved to evade this response and go systemic as infection progresses. And so eventually it does make its way into the blood and to secondary lymphoid organs such as the liver and the spleen. And this constitutes basically the systemic spread of the pathogen. And so, you'd all know that Salmonella commonly causes a typhoid fever in humans. In mice, the model pathogen that we use is Salmonella typhimurium. It's essentially about 99% similar to Salmonella typhi, but but these pathogens are host restricted. That is, typhi infects only humans, causing very limited gastroenteritis in mice, and typhimurium does the opposite. That is, it causes systemic disease in mice, but causes very self-limiting gastroenteritis or food poisoning in humans. And so, most of us have had Salmonella infections, and no, it doesn't feel good, but we also know that we resolve Salmonella typhimurium rather spontaneously. But mice on the whole don't. And so mice are the model systems that we've been using to model Salmonella typhimurium infection. So Salmonella is quite a clever pathogen. And like most host-adapted pathogens, Salmonella actually switches its gene expression in response to the host environment. And most pathogens will do that. They don't express all virulence genes all the time. They actually express them within the right compartments or the right environment of the host to be able to prevail in that environment. And so early on in the gut lumen, Salmonella is largely extracellular, and it upregulates expression of SPY1, which is basically Salmonella pathogenicity island 1. And Salmonella has two pathogenicity islands, SPY1 and SPY2. So SPY1 is up in the early phase of infection in the intestinal lumen. And as infection progresses and Salmonella becomes intracellular at more systemic sites, it starts to upregulate SPY2. But mind you, this is a gradual process. And so, it's some of the genes are switching phenomena, but on the whole, the induction of SPY2 is a gradual process, and it'll happens over time as Salmonella becomes systemic. And in mice, we can actually model this whole process in about 5 days using an IP model. We can also model this phenomenon in a very limited macrophage system, basically, where we just infect macrophages with Salmonella and follow gene expression of bacteria over time. And we can see that early on, Salmonella turns on SPY1, and then later on, as it becomes intracellular inside macrophages, it turns on SPY2. And so this in vitro macrophage infection system is one that we've been using in the lab, coupled with the in vivo IP model of Salmonella infection. And so one example of this switching in gene expression is provided actually by bacterial flagellin. And so flagellin early on within macrophages is sensed by this protein called NLRC4. It is one member of the NLR family that I spoke about a little while ago. And this leads to activation of Caspase-1 and cell death and production of IL-1 beta, neutrophil recruitment that leads to clearance of the pathogen. Again, this was an axis I just described in my introductory slide. And as infection progresses, Salmonella has actually evolved to downregulate expression of its flagellin and actually evade recognition by NLRC4. This leads to reduced pyroptosis, intracellular replication, and actually intracellular survival of the bacterium, which enables its evasion and its dissemination. So these macrophages are cargo cells that can carry it to systemic sites. And so, it was known for a long time that Salmonella switches from this flagellin high to flagellin low phenotype, but how it actually does that was not entirely clear. And so in recent work, we focused on this, and we actually found quite an unexpected role for the NLRC4 inflammasome and for caspase-1 in actually regulating expression of its own ligand, that is flagellin. And so in a gist, what we found is that activation of NLRC4 and caspase-1 and the accompanying cell death is accompanied by release of this host lipid called Atpase, which can actually signal back to Salmonella and increase expression of its flagellin. And as infection progresses of host cells over time, the host actually produces type 1 interferons that actually dampen this whole axis, essentially. And this, so we think this is actually the host has evolved this mechanism as a protection, basically, right? Because you don't want every single one of your cells to pop dead through NLRC4 activation. And so the host has to shut down this axis to avoid immunopathology. But what we find is that Salmonella actually takes an advantage of this NLRC4 and caspase-1 low and cell death low environment to actually survive intracellularly, evade innate immune detection, and disseminate to higher bacterial loads systemically. And so, this was quite a self-contained study. But as we know, cells and organisms are actually complex networks, and so is Salmonella and its corresponding host. And we're recognizing that, of course, there are multiple interactions that happen between the host and the pathogen that actually determine how a pathogen actually hides away inside the host cell. And so, together with the Baliga lab at ISB and Eliza, Mario, and Amardeep in Nitin's lab, Ajay in my lab has been leading this project, and he went ahead and he actually sequenced Salmonella, intracellular Salmonella, from wild-type macrophages over time. And basically what we did here is we collected the bacterial transcriptome. We can actually enrich intracellular Salmonella, which, as you would imagine, are very few in number. These are intracellular bacteria. So we enrich them with specific baits, and then we sequence them, collecting about 8 million reads for the bacterial transcriptome. And in conjunction, Ajay also sequenced the host cell that was housing the Salmonella, and generated data on the host transcriptome. But today I'll be focusing mainly on the pathogen sequencing aspect of it, which is sequencing of the bacterial transcriptome. And so I told you a little while ago that we found that caspase-1 can actually regulate production of its own ligand. And so we reckon that something more complex might be happening in the presence of caspase-1. And so when we initially got into this pathogen sequencing, we decided to sequence bacteria from both wild type and the caspase-1 knockout environment, and see if we can recapitulate existing features that we'd already reported and find new ones. Yeah. Right. And so I think I missed the model system that the pathogen that you're working with, or in case you mentioned that, but I suppose it's immaterial. So basically our model system is bone marrow derived macrophages. So we isolate macrophages from the bone marrow of mice. We differentiate them in vitro with MCSF for 5 days, and then we use those to infect with Salmonella. Now, macrophages actually undergo very rapid cell death upon Salmonella infection within about 30 minutes, but not all macrophages die, right? And so there are two things here with regard to the distinction between innate and adaptive immunity. One, I would say, is the time points that we're looking at. So most of the data I'm going to present to you today, actually all of it with regard to the pathogen sequencing is at 6 hours post Salmonella infection. The second is that this is a self-contained macrophage system, and so we don't really expect any contribution from adaptive immune cells here. So yeah, does that answer your question? All right, I'm guessing it does. You guys can still hear me, right? Alexander, can you still hear me?
Let's move on.
Yeah.
Yeah.
Thank you.
Okay, perfect. So when we sequenced the intracellular Salmonella, we went ahead and we did a differential gene expression analysis between genes that are expressed in Salmonella that is present in wild type macrophages compared to just the inoculum, that is Salmonella, that was used to originally infect these macrophages. And then we also did a differential gene expression analysis between the wild type bacteria that are housed in the wild type and the Caspase-1 knockout macrophages. And so I should mention a word of caution here. The Caspase-1 knockout macrophages that we're using are actually 111 double knockouts. And this is, the field in 2012 realized that all Caspase-1 single knockouts were actually double knockouts because 1 and 11 are not far enough in the genome to be separated during homologous recombination. And so later on, mice were made that reintroduced Caspase 11 into the Caspase-1 knockout. But so I want to put that out there, and I will clarify the specific role of 1 and 11 a bit later in the talk. So when we sequence these intracellular bacteria, we find that we can recapitulate what we expect to say, basically. So, we see that at about 6 hours post-infection, Salmonella that is housed intracellularly within macrophages. So mind you, all extracellular bacteria here have been killed with gentamicin, and only the intracellular bacteria are being sequenced here. And we can find that in wild-type macrophages, Salmonella downregulates genes that are comprising Salmonella pathogenicity island 1, and it actually upregulates genes that are part of Salmonella pathogenicity island 2, essentially. So these are the genes that are required for its intracellular survival. We also had, like I said previously, shown that Caspase-1 actually maintains expression of flagellin on Salmonella. And so the expectation there is that in Caspase-1 knockouts, flagellin would actually be downregulated faster, which is also what we'd shown in our previous paper. And so we looked for two genes, the gene that encodes flagellin, that is FlyC, and FLHD, which is basically the master regulator of the flagella operon. And basically what we find is with the wild type in solid lines and the 111 knockout in dotted lines, that in the 111 knockout, FlyC, which is in the black lines, is downregulated faster in the 111 knockout, and then FLHD, which is the master regulator of the flagella operon, is also downregulated faster in the 111 knockout. So kind of recapitulating what we'd seen previously and giving us some confidence that we can trust what is going to come out of this sequencing data. So we then cast a broader net to ask, all right, what else is going up and down in these intracellular bacteria in these two different host environments, that is the wild type and 111 knockout macrophages. And as expected, we see genes related to flagella assembly downregulated in the 111 knockouts. And quite interestingly, we found that the 4P4Q regulatory system, which is essentially a two-component system, in Salmonella and also in some other enteric bacteria, is actually upregulated in the 111 knockout. And so this was quite striking to us because the 4P4Q regulon can really have wide-ranging effects on Salmonella physiology. So quickly a word on the 4P4Q system. So basically, the job of the 4P4Q system is to modulate gene expression according to the environment, essentially. And 4Q is actually the sensor kinase that is activated in these stressful conditions in the Salmonella containing vacuole inside cells. So these include things like low pH, low magnesium, the presence of host antimicrobial peptides, and 4Q actually then phosphorylates 4P, and 4P induces expression of a bunch of genes that are necessary for Salmonella to survive inside the host. And this includes many genes, including in the induction of genes that are involved in imparting antimicrobial resistance. Antimicrobial resistance. So I mentioned this here as CAMP resistance, and CAMP essentially stands for cationic antimicrobial peptides. And a bunch of 4P-induced genes actually confer resistance to host antimicrobial peptides. Among them are some listed here, and also PAG-C and PHO-N, which I'll come to a bit later. And so we then asked, okay, we see induction of this 4P regulon intracellularly to a greater extent in the Caspase 111 knockout macrophages. So how about the genes that map to the 4P regulon? And indeed, we can see that in the 111 knockout macrophages, which are in red here, compared to the wild type, which are in black, Salmonella actually upregulates expression of all of these genes that are part of the 4P4Q regulon, essentially. They're induced by 4P4Q. And so we went ahead and we made reporters for two of these genes, PAG-C and PHO-N, in collaboration with Kelly Hughes, who's essentially a master bacterial geneticist at the University of Utah, and the collaboration with him has really facilitated our study. So big shout out and thank you to him. And he constructed these PAG-C and PHO-N reporters of Salmonella for us. And this allowed us to query at the single cell level in situ what actually happens to the expression of these 4Q dependent genes in the wild type versus the 111 knockout environment. And so in red is basically a constitutive gene, which is LPS in this case. In green, you can see PAG-C, and hopefully you can appreciate that bacteria that are in the 111 knockouts turn on PAG-C to a much greater extent. And then the third panel is basically a merge or an overlay. And we see this not only with PAG-C, but also with PHO-N, which is another 4P4Q inducible gene. Like we see with PAG-C, it's induced to a greater extent in the Caspase 111 knockouts. And so I told you that 4P4Q is actually involved in imparting resistance to host antimicrobial peptides. And these, host antimicrobial peptides are an important host mechanism to defend against intracellular pathogens. And one of the antibiotics that is commonly in use, that is polymyxin B, is actually of a very similar cationic nature as these host antimicrobial peptides. And essentially, it can it can bind to the lipid A portion of the LPS, and it can alter bacterial membrane permeability, and that's the way it kills bacteria. And Salmonella has evolved multiple resistance strategies to polymyxin B and antimicrobial peptides that are imparted by activation of 4P4Q, some of which are listed here just for the Salmonella aficionados. And so then we asked, all right, if there's elevated 4P4Q induction in the Caspase 111 knockouts, does this actually affect antimicrobial resistance of Salmonella that is housed intracellularly in the 111 knockout environment? And the answer is that it does. And so we tested these two host antimicrobial peptides, C18G and PG1, and then we also tested polymyxin B, which is actually a last line drug against gram-negative bacterial infections. And we can see that bacteria isolated from the 111 knockout environment are actually resistant to these host antimicrobial peptides and also to polymyxin B. And as a little bit of detail, the growth in the absence of cationic antimicrobial peptides is set here at 100%. So any decrease that you see is basically a percentage decrease in growth isolated from the wild type bacteria, and any recovery that you see is again a percentage recovery here. So it appears that bacteria isolated from the 111 knockouts not only induce more 4P4Q, they're also resistant to these antimicrobial peptides. But this is seemingly specific to polymyxin B and to these host antimicrobial peptides, because you don't see this across the board with all antibiotics. And so there's no difference in resistance of these bacteria housed in either wild type or 111 knockout cells to, for instance, gentamicin or ampicillin. And so we next basically asked, is this antimicrobial resistance actually dependent on 4P4Q? And to do this, Kelly Hughes actually constructed a few strains for us. So he made a strain that is 4P constitutive or that is deleted for 4P. And we use these two strains to infect both wild type as well as Caspase 111 knockout PMTMs. And the color scheme here is the same. The wild types are in black. The 111 knockout bacteria are in red. But this graph is a little more involved. So let me just take you through this. And if you just follow the arrows. And so hopefully what you can appreciate is that compared to the absence of any polymyxin B or antimicrobial peptides, bacteria that are housed in the wild type environment are actually susceptible to polymyxin B. Bacteria that are housed in the 111 knockout environment are resistant. But now, if you have constitutive 4P on the wild type environment, basically, that is, these bacteria actually can't downregulate 4P. They are constitutively on for 4P in the wild type, and so these are now resistant even in the wild type environment. And bacteria that are actually knocked out for 4P, which is basically this strain, is susceptible in the Caspase 111 knockout environment. And so what this indicates to us is that indeed this increase in antimicrobial resistance that we see in the 111 knockout environment is mediated through induction of the 4P, 4Q regulon and Salmonella. So any questions at any time, please feel free to interrupt me, just in case anything isn't clear. So whatever I talked about right now is on the bacterial side. And so we went ahead and asked, what about the host side, basically? How does actually host Caspase 1 or 11 mediate this phenotype? And before getting into that, I should remind you that Caspase 1 is actually an enzyme that is activated by proteolytic cleavage as a result of assembly of this inflammasome. And so the NLRC4 inflammasome upon assembly causes cleavage of pro-Caspase 1 into active Caspase 1. And this phenomenon is important for inducing cell death called pyroptosis and release of these inflammatory cytokines. And so the first question that we asked was, is the effect that we're seeing dependent on Caspase 1 or Caspase 11? Because remember, all the data I showed you so far was in the 111 double knockouts, which lack both genes. And so now we're using a Caspase 1 single knockout. And basically what we find is that the Caspase 1 single knockout behaves identically to the 111 double knockout, which kind of indicates that the absence of Caspase 1 is sufficient to impart this resistance to antimicrobial peptides to Salmonella. But we know Caspase 1 is actually activated as part of this inflammasome, right? And so we next went ahead and just systematically asked, what about the other upstream inflammasome components? Are they involved at all? And so we use macrophages that are knockouts for ASC, NLRP3, NLRC4. These are all NLR components that have been inflammasome components that have been involved in Salmonella detection. And again, we use the Caspase 111 double knockouts, and we also use the Caspase 11 single knockout. And the gist essentially is that none of these upstream inflammasome components is required for imparting this antimicrobial resistance. We see this phenotype in this only in the Caspase 111 knockout and only in the Caspase 1 single knockout. And so because we knew that Caspase 1 is activated in response to its cleavage, we asked, what happens if we inhibit activity of Caspase 1, right? Because everything that we see in the literature, or most of it, that is reported for Caspase 1 is dependent on its proteolytic cleavage. That is its activity, because the inactive pro-Caspase 1 is basically believed to be a precursor that kind of sits around in the cell waiting to get activated in response to pathogens. And here we were seeing a phenotype where Caspase 1 was imparting this, lack of Caspase 1 was imparting this antimicrobial resistance. So we went ahead and we used an inhibitor for Caspase 1 activity. And we've used two different inhibitors. I'm showing you the data here for only one of them. And basically what we find is that inhibition of caspase-1 activity, again, has no impact on polymyxin B resistance. And so if you inhibit caspase-1 activity, bacteria isolated from activity-inhibited macrophages or wild-type macrophages behave identically. And this is just data to show you that an inhibitor actually works. And so we inhibit caspase-1, we actually don't see production of IL-1 beta like in the caspase-1 knockout, which is what we would expect essentially. And we went ahead and then queried this in situ at the single cell level. And again, we use this 4N reporter with a constitutive M-Cherry reporter. And basically, again, what you can see is that the bulk data is recapitulated in single cells where bacteria housed in the 111 double knockouts have higher induction of 4N. But regardless of whether or not you inhibit caspase-1 activity, this does not change in the wild type environment. And so it appears that this phenotype that we're seeing of higher 4p4q induction is dependent on caspase-1, but it's independent of caspase-1 activity. And in some very preliminary data, we have some idea as to how this might be happening. I would not go to town with this just yet, because we want to do this at least in another 2 or 3 different ways. But essentially what we think is that this increased induction of 4p4q and polymyxin B resistance is dependent on the vacuolar pH. Because if we neutralize vacuolar pH with treatment of by treatment with bafleomycin, basically we lose this phenotype in the 111 knockout. So bacteria isolated from BAFE-treated caspase-1 knockout macrophages actually show induction of 4N to similar levels as the wild type. And so it appears that there perhaps is a higher vacuolar pH of the caspase-1 knockout macrophages that is imparting this a higher 4p4q induction in Salmonella. And we can also see this with resistance to polymyxin B. So you lose 4p4q induction by bafleomycin treatment of the 111 knockouts. And if you look at this last bar here, you lose polymyxin B resistance of bacteria isolated from the caspase-1 knockouts if you treat with bafleomycin A. And so basically we've now gone ahead and in relatively preliminary data, we've gone ahead and tested in vivo whether this phenotype actually recapitulates. And basically here what we did is we infected mice intraperitoneally with 200 CFU Salmonella, and at day 5 we isolate the spleens and we look at the adherent CD11b positive splenocytes. We measure expression of the 4N reporter, and then we also measured growth in polymyxin B. And hopefully what you can appreciate here, we're working on quantifying this data across multiple mice and multiple fields, but it's a pretty all or none phenotype. And so I feel relatively comfortable telling you that we can see in vivo that bacteria that are isolated from adherent splenocytes on day 5 in the caspase-1 knockout environment have higher induction of 4N compared to those that are isolated from wild type mice. And if this is preliminary data, so we've done this across 3 mice per genotype at this point. And we went ahead and, in conjunction with 4N induction, we also cultured these intracellular bacteria in polymyxin B. And again, what we find is that bacteria isolated from intracellular bacteria isolated from the 111 knockout are actually have higher resistance to polymyxin B compared to those isolated from wild type mice. And so, we're still working on this. This is still work in progress, but a working model right now is this, which is that in wild type macrophages early on, we know that Salmonella activates the analyze C4 inflammasome. This leads to activation of caspase-1 from its inactive precursor, and this leads to cell death. And so, this does happen early on, but we also know that Salmonella survives intracellularly within macrophages. And so what we're beginning to think is that, so obviously not every host cell dies, and Salmonella has a way to prevent that. The host has also evolved ways, especially type 1 interferons that I was talking about earlier, to actually shut down this active cell death process, right? So, what we think is that in the of course, a set of macrophages die early on in response to Salmonella infection, but in the subset that survives, there's actually an activity-independent role for caspase-1 in somehow neutralizing or inhibiting, let's say, or changing the pH of the vacuole that actually dampens this whole axis, essentially. So normally, Salmonella would turn on induction of its 4P4Q system inside macrophages in response to the acidic pH, and through this gene called PMRA, become resistant to host antimicrobial peptides, which would allow it to survive inside the macrophage. And we think that caspase-1 in an activity-independent way kind of inhibits this whole process and allows bacteria, facilitates bacteria to survive inside macrophages. And of course, if you lack caspase-1, which is all the data that I showed you, this basically does not happen, and you get higher resistance to antimicrobial peptides. And so, why are we excited about this? I think conceptually, I'm excited about a possible what I call non-canonical, but it's not a usual role for caspase-1. It's activity independent. And we've normally thought of caspases as precursors that are poised to get activated in response to danger signals or pathogens. And most of the reported phenomena that are out there are dependent on caspase activity. And so it appears, though, from this data that there are other collateral functions for these inactive caspase precursors in regulating the physiology of intracellular bacteria. Evolutionarily, we think that this axis in terms of where caspase-1 actually shuts down antimicrobial resistance of Salmonella and makes it more susceptible to antimicrobial peptides, that this axis might have evolved to actually might have evolved in the host in order to make bacteria more susceptible to antimicrobial peptides. But because polymyxin B actually mimics these antimicrobial peptides, we see that same phenomenon with polymyxin B as well. So evolutionarily, we think that in the subset or surviving cells, this is a host strategy for pro-caspase-1 to actually inhibit antimicrobial resistance of bacteria and make them more susceptible so that the host can actually clear these intracellular bacteria. Any questions on this? All right, so I assume that was either totally clear or totally opaque, but feel free to jump in at any time.
I think there's some questions in the chat. If you wanna, I can read it out.
Okay, yeah, let me find.
One of them is asking about whether there's evidence that Salmonella inhibits the caspase pathway.
Oh yes, I see.
There's evidence of expression downstream being suppressed.
Species inhibiting the caspase pathways, the caspase-1 may be expressed, but is there evidence of expression downstream being suppressed? Secondly, not all cells die, which may be physiological, although perhaps Salmonella species. Yeah, and so the answer is yes. Salmonella does inhibit. Well, so Salmonella itself has not evolved to, at least we haven't found data that Salmonella can inhibit the caspase-1 pathway. The inhibition that I was talking about is more host mediated. You are right, that is quite a possibility that Salmonella would like to inhibit caspase-1, right? Because activation of caspase-1 actually causes Al1 beta production, neutrophil recruitment that leads to clearance of Salmonella. And so there is a reason for Salmonella to actually actively to actively inhibit this process. But we haven't so far found evidence of it. I mean, given the arms race between the host and the pathogen, it appears feasible that Salmonella may do so. But most of what we found indicates that the host actually shuts down this caspase-1 axis as a means to prevent ongoing host cell death and prevent immunopathology. Okay, so thank you for that. Judith, do I have about, how much more time do I have?
So
I got one more
we have
question.
one more, yeah, sure.
Could I ask you a question? Do we
Of
have time?
course, yes.
So I'm not an immunologist, but I wanted to understand the difference between apoptosis and pyroptosis. Yeah, this
Yes.
other version of programmed cell death. I'm like, what is like the most important feature here that's like preventing the spread of infection of Salmonella compared to like apoptosis? Like if it triggered apoptosis instead, would it work, or would that be bad? Or
Yeah, so it's a good question. So basically, pyroptosis is cell death that is dependent on caspase-1. Apoptosis is normally dependent on it also involves caspases, but it's dependent on other caspases, such as caspase-3 and caspase-9. The lines between pyroptosis and apoptosis are rapidly blurring, at least in the context of Salmonella infection. And there's now believed to be, for instance, involvement of caspase-8 early on in pyroptosis. Caspase-8 was actually classically thought of as an apoptotic caspase, essentially, right? And so basically, pyroptosis and apoptosis are also what's the right way to say this? They're time dependent during Salmonella infection, at least of macrophages. And so early on, you get pyroptosis within 30 minutes, essentially, at least in cell culture. And in vivo, we know pyroptosis is important for controlling bacteria. So mice that lack caspase-1 actually fail to resolve Salmonella infection. They have much higher bacterial loads. Apoptosis also occurs, but it occurs later in infection. And mice that lack apoptotic caspases are not going to, apoptosis is believed to be not as important as pyroptosis. Let's just put it as that. So it's not going to be self-sufficient in controlling Salmonella infection, but there is evidence that when you lack pyroptosis, that is when you lack caspase-1, that Salmonella then starts to preferentially induce apoptosis in macrophages, but that is not self-sufficient for controlling the infection.
Okay, thank you.
Okay, thank you for the question. Okay, so Judith, how much more time do I have?
I think about 10 minutes or so.
10 minutes or so. Okay. Yeah. So I've taken a lot of time on the first story. And so I wanted to talk to you a little bit about some of the work that we're doing on Lyme disease. And so what I'm going to do is I'm going to skip over a bunch of slides and cut to the chase. So basically, what is Lyme disease? It's this vector-borne illness caused by Borrelia burgdorferi. It's transmitted by tick bite, and the risk of infection to humans is the highest during the summer months. It's mostly prevalent in the northeastern United States, but it's also prevalent all over the world. And most of you would know that it actually manifests as this characteristic bullseye rash at the site of infection. And so this bullseye rash is actually the site of, is also the site of the immune response. And let's skip over this. And so what are the challenges in studying Lyme disease? Well, basically, one is that most of the studies have been done in mice, which are actually reservoir hosts. And so they actually don't adequately recapitulate the human immune response because they don't develop the disease like humans do. And also laboratory strains of Borrelia really differ widely from Borrelia strains that infect humans in the wild. Actually, Borrelia strains that are laboratory adapted have lost a bunch of plasmids that are required for virulence. And the human studies are also really static and narrow in scope, the ones that are out there. And so let's just skip over this introduction and cut to the chase of what we actually went after. these introductory slides were mainly to tell you that the immune response happens at the site of the skin, that the tick saliva can actually suppress some of the skin immunity, and Borrelia is highly motile. So when it can migrate through the blood vessels through these endoflagella and disseminate to joints and just cause a wide variety of problems, essentially. And finally, that a subset of individuals never actually resolve the disease, and they progress to what is called post-treatment disease. And so our approach to studying this has been to recruit a prospective cohort of individuals and follow them over time. From diagnosis out to 1 year. We collected data at multiple scales, circulating mediators on the cells in their blood, and also metabolomics. We integrated this with data from the site of infection and with clinical information that was collected in these individuals, with the goal of trying to get a handle on what's happening during the human immune response to Lyme disease. And so, Borrelia causes just a wide variety of symptoms across individuals. And so you can see that most of these symptoms occur at the time of diagnosis. And by about 6 months, which is the green and the blue bars, most of these symptoms are actually resolved. And at the time of diagnosis, people go on antibiotics, and that helps to resolve some of these symptoms as well. Now, the circulating immune protein signature in the blood actually follows this exact pattern, which is at the time of diagnosis, we can see upregulation of a unique signature, unique set of proteins in patients compared to controls. But much of this actually resolves by about 2 weeks, which is T2, and then 6 months and 1 year post-antibiotic treatment. So the circulating immune protein signature also occurs primarily at diagnosis. We then went ahead and asked, all right, what happens if we look at the circulating immune protein signature stratified by symptoms in individuals? And we can uncover basically 2 signatures here. One is this interferon gamma, predominant interferon gamma signature and interferon gamma inducible genes like CXCL 9, 10, 11 that actually correlate with the symptom severity, which is pretty, so interferon gamma and interferon gamma inducible proteins are very high in the plasma of individuals. And then secondly, we kind of uncovered this circulating signature of what appears to me to be more of a type 2 immunity kind of skin signature that correlates with the dimension of the erythema migrans and the number of erythema migrans. So some people, the size of the bullseye rash and then the number of bullseye rash. And some people have it, like one individual in this study has as many as 35 bullseye rashes. So almost like fell into a tick bush or something. And so these appear to be the gamma and the skin signature appear to be 2 predominant signatures that we pick up. And we can see that these proteins actually in the plasma kind of follow 2 trajectories. One is a set of proteins that falls very rapidly after antibiotic treatment. And then a second, the blue set of proteins are actually proteins that fall slightly, but actually don't resolve completely until later, which is about 6 months in infection. And if we do modeling based on these proteins, we can actually develop a classifier of about 5 proteins that can classify patients from controls at the time of diagnosis and at about 2 weeks post-diagnosis with about 70% accuracy. Now, this isn't great, but considering the fact that, we don't actually know there are many challenges associated with Lyme disease study, right? People don't know when they actually got the tick bite. So we don't completely know what phase of the immune response they're at when they come into the clinic and so on. So, the sets of challenges with this human data is immense. And I won't go into all of them, but happy to discuss it offline if anybody's interested. So then if we look at the cells in the blood, and this is just flow cytometry data that was collected on about 45 different immune cell populations. Basically, the gist here, without going into all of the data, is that there is just a striking lack of peripheral changes during Lyme disease. And so, you can see here that in controls, as well as in patients, patients are really clustering largely by identity rather by whether or not they have Lyme disease, essentially. And so the inter-individual heterogeneity appears to really be the largest driver of changes that are occurring in peripheral immune cells, except for this one patient sitting here at the top. This is the person who had 33 EM rashes. And so it appears that, when you really have severe disseminated disease, you can see a response in the periphery. But, we have people in this cohort, even with 24 rashes or 10, 15, just an odd number of rashes, and none of them show anything in the periphery. Apart from this individual. So kind of reinforcing this stealth nature of Borrelia, this is again data that we're thinking through. But it appears that Borrelia, for the most part, at least early on, is evading this peripheral activation of immunity. There are some small changes in plasmablasts that are consistent with prior literature. So plasmablasts are upregulated at diagnosis. And in the person that has 35 EMs, we can see induction of a CD4 effector T cell response of some non-classical monocytes being elevated. That person is shown as a red dot here among the other patients. And, also seemingly an induction of gamma delta T cells, although, I'm not totally convinced of that given what these box plots look like. We can also show this as this radar or circus plot, and the person with 35 EMs here is in red. And again, you can appreciate that apart from this individual, most individuals actually hardly show any changes in immune cells in the periphery. And this individual also has the highest circulating immune protein signature. So what about that signature, right? I mean, I told you we see a circulating immune protein signature rich in interferon gamma and inducible proteins, rich in, what appears to be more of a type 2 signature. So where is that coming from? And so we did to cut to the chase, we basically did a meta-analysis of both PBMC data and skin data from the literature. And basically what we find is that circulating immune protein signature in the plasma maps back to the skin. And this is just single cell RNA-seq data. And you can see that these are all skin cells, keratinocytes, fibroblasts, and endothelial cells, myocytes. And most of the cytokines that I was talking to you about, the IL-18, CXCL, 9, 10, 11, these are all upregulated in these different types of skin cells, not so much in the periphery. And we can also then look at myeloid cells, which are producing, most of the IL-10 and the CXCL8 and most of the interferon gamma, which was the predominant signature, again, appears to be coming from the skin, but from the T cells and the NK cells that have infiltrated the skin in response to the disease. And so, there's obviously a lot of other research that needs to be done on this cohort. We're working through single cell RNA-seq data and metabolite data to see how those pan out. If we see the striking lack of immune activation, I should say in immune cells in the periphery, we're hoping the single cell RNA-seq on the PBMCs will give us some idea about, is there just not anything getting activated in the peripheral cells, or is there some kind of inhibitory signature that these cells are expressing? Those are all things that we're currently investigating. But in a gist, we see that we can pick up a circulating immune protein signature that can moderately discriminate patients from controls. This includes a type 2 interferon response that associates with disease severity. But in spite of the circulating signature, there's really minimal activation of immune cells in the periphery. And most of the circulating proteins in the blood actually originate from the site of infection, except for this one person with severe disseminated disease, where we can see, marked changes in immune cell abundance. So, it appears that Borrelia, for the most part, is escaping this peripheral immune activation. And one of the reasons why why I suppose we find this interesting is We know a subset of individuals never resolve Borrelia infection and move on to develop post-treatment disease. And we don't completely understand why that happens. We know that antibodies do develop against Borrelia, and T cells, can be active. So antigen-specific T cells can be detected in the periphery in T cell and B cell sequencing studies. But at least at these early time points, unlike other infections that we've studied, COVID, influenza, Salmonella, even psoriasis, basically, which is a skin originating infection, but more, of course, skin originating disease, infection independent. Borrelia just appears to be this pathogen that kind of doesn't do anything in the periphery. And we wonder whether this lack of peripheral immunosurveillance may be the reason why some people actually escape a robust immune response and then go on to, can't control Borrelia in the periphery and then go on to develop issues like neuroborreliasis, where the pathogen, through the blood can get into these multiple different types of organs like the brain, the heart, and the joints, and so on and so forth, and impact multiple organ systems. So I will move now straight to my acknowledgement slide. I was hoping to talk about chronic Lyme disease, but I think that is not necessary. And this was all future work. So with that, I'll just thank everybody who was involved in the work. Most of the Salmonella work was done by Ajay. I mentioned most of these people along the way. All of the Lyme disease work was driven by Cliff, just an enormously talented research associate in my lab without a PhD degree, has just moved on to do a PhD in genome sciences, but did all of the work right from the experimentation to the modeling and so on. Big shout out to Kelly Hughes, who's helped a lot with bacterial genetics on the Salmonella front. Nitin Baliga, who's helped with pathogen sequencing, and our collaborators, Gary Wormser, who's a clinician that helped us acquire the Lyme disease cohort, and just a variety of collaborators at ISB who've made this complex study possible. So with that, thank you for listening. Sorry for going over time, and happy to take any questions that you may have. I'm looking at the chat box. So there's a question from Maria about the localization of VATPase and the Salmonella system. So we looked at VATPase. We don't see any differences at all. Basically, that was one of the things we thought about. That could Caspase-1 be actually inhibiting vacuolar acidification through the VATPases? There is no change there. Little more technical, could you tell us which species of S-type immunity was used? So we're using the SL1344 S-type fumarium. MRA also wanted to know, when we did the enrichment for intracellular Salmonella, was the enrichment for total bacterial RNA or a subset? So the enrichment was for mRNA, basically. So we use baits. Essentially, these are baits that actually are mapped to the Salmonella genome, and then they are used to pull down the Salmonella mRNA. So they're biotinylated baits. And I would encourage you to go back and look at Nitin Baliga's pathogen sequencing paper. They actually reported it originally with Mycobacterium tuberculosis, where they engineer baits to the Mycobacterial tuberculosis genome to actually pull down intracellular bacteria, and we adopted it to Salmonella type fumarium. Actually, I think they did it with Mycobacterium in vivo, essentially. So their loads were slightly higher, so to say. But yeah, essentially, there are baits that map to the entire genome. So Georgie says, just a random thought, could it be that this large difference between the 35 EMs patient and the others in the cohort is due to an evolved strain of Borrelia burgdorferi or this is not possible and we're better off carrying research on the host? So it's a great question. Again, I didn't go into all the complexities of this Lyme disease data, but you're right. It could be the strain of Borrelia burgdorferi that happened to infect this individual. It could also be that this person just has a genetic variant that makes this person just, more susceptible to mounting a huge immune response. In this case, it would not be a bad thing, so to say. But this person also had disseminated disease, right? So, it's the answer is not clear, basically, right? It could be something in the pathogen, or it could be something in the host. But I think the thing to really take away from this is that, even if we say that this response that we're seeing in the person with 35 EMs is a true response, right? It's an N of 1, and so one has to take that with a grain of salt. But even if that is a true response, I mean, there's an enormous thresholding effect here where, right up to having about 25 to 28 EMs, where people clearly have disseminated disease, we're not seeing anything in the periphery. And so I think that to me is the really striking thing here, that, this pathogen just has a way of silencing these classical monocyte activation, the NK cell activation, and so on and so forth, that we would classically pick up in the periphery for many other bacterial infections. Happy to take any more questions if there are any, but I know I'm over time.
Yeah, just one, because I was going to ask you actually whether you see a switch that for people that develop sort of chronic Lyme disease, whether then there is a systemic signature.
Yeah, Judith, that's a good question. Man, chronic Lyme disease is its own beast. And, I'm so sorry that I skipped over all of those last slides. And so I don't have the answer to that. And, the studies that are out there on chronic Lyme disease are very limited. Most of them have actually been done by clinicians. There are no systems level studies on chronic Lyme disease, but the bigger problem in the field really is, what is the definition of chronic Lyme disease, right? I mean, I think that is what that is what the field right now is struggling with. And so, it is interesting, and certainly a great hypothesis that we may see this persistent signature in chronic Lyme disease. The one thing that we want to do for we call it persistent Lyme because chronic is just riddled with, the word chronic is riddled with so many controversies in the field of Lyme disease. Post-treatment Lyme disease, where people actually get antibiotics and then go on to develop Lyme disease, persistent symptoms is much better established. So, but much in these chronic patients, what is called classically chronic Lyme disease, Judith, is basically individuals who never get diagnosed, essentially. So the bullseye rash occurs in about 70% of the individuals. About 30% of the people never show the bullseye rash, so they never get to the clinician in time, and they land up in the clinic with all of these systemic problems like facial paralysis and things like that. And then if they test positive for Lyme, diagnosis in the Lyme field is also a big problem, which is why we're hoping that some of these early immune-based biomarkers may speed up diagnosis. But in a gist, the clinical definition is a problem. Who has chronic Lyme? The C6 test that is used to diagnose Borrelia infection can also fluctuate between being positive and negative, because Borrelia changes its expression of this VSLE peptide as it transitions through the host. And so people will test positive, and then they come in 2 weeks later and they test negative, and it's and they never showed the bullseye rash in the first place. And so clinicians are just they're like, do you have Lyme disease? Do you not have Lyme disease? We don't know that. So anyway, sorry for such a long answer, but what we hope to do is actually recruit a cohort of people who have chronic Lyme disease as defined by clinicians. That is, you can pick up antigen specific responses to Borrelia in the periphery and so on. So we're coming up with a definition for this, what we would actually define as a chronic Lyme disease cohort. And then the first goal here really is to just follow them out and do a molecular stratification, right? So to kind of develop good definitions of the disease, of course, based on the immunology and the other omics types of data. So I think, that's where chronic disease lies. And sorry for that little detour in response to your question.
No, that's fascinating. Thanks so much. I think we'll leave it there. Thanks again for giving a talk. And I think you have a couple of meetings lined up after, right?
Yeah. Thank you so much. Thanks everyone for listening.